Hello, and welcome to today's session on clinical trials with tobacco products, examining the abuse potential, exposure, and biomarkers for tobacco. My name is Beatrice Setnik, and I'm the Chief Scientific Officer of Altasciences. Today's learning objectives are to discover more about the clinical trials required to support market applications for tobacco products in the United States. We'll talk about the discovery of the clinical trial methods to evaluate the abuse potential, pharmacokinetics, and smoking cessation. We'll also examine how drug administration, dosing, and selection of study subjects are handled in these types of trials, and discuss the relevance of examining biomarkers in clinical trials. So just to give a few commonly used terms that you'll hear throughout this presentation, a tobacco product is any product that's made or derived from tobacco that's intended for human consumption. And that includes any of the accessories or parts of that tobacco, except for the raw materials that are made in the manufacturing process. This also includes rolling papers and filters. An electronic nicotine delivery system will be often abbreviated throughout the presentation as an ENDS. These are devices that heat and deliver nicotine, and you see a few examples there on your right-hand side. Modified risk tobacco products, or MRTP, are tobacco products that have innovative changes that substantially reduce or eliminate either or both their toxicity and addictiveness of the product. And we'll be talking a lot about those and how those are evaluated in clinical trial settings. So it starts with the guidances for industry that the FDA has issued. There are 3 of them that particularly hone in on new tobacco products. So that is any new tobacco products that has been issued. This guidance addresses the addictiveness of the product. We also have a specific guidance for the ENDS, which is the electronic nicotine delivery system, and for the MRTPS, which is the modified risk tobacco product application. And the common theme to all 3 of these is that they all address and need to address the abuse potential or addictiveness of the products. There are also sections in all of those guidances that address the need to measure biomarkers, and we'll go into more detail about what type of biomarkers we're looking at, not only nicotine exposure, but biomarkers of harm or exposure. So we'll be talking about that, and that applies to all tobacco products, including the ENDS and the MRTPs. So we'll talk about first the evaluation of abuse potential and how that's done in a clinical setting for tobacco products. The goal with modified risk tobacco products is to eliminate the human abuse potential. That is primarily driven for drugs. We're trying to reduce drugs to prevent them from being eliminated. When you have a modified risk tobacco product in which you're intending to switch smokers, heavy smokers, over to a modified risk tobacco product, the abuse potential is not going to be eliminated. If there was, the smokers would not likely be switched to that product. So the abuse potential needs to be somewhere lower than the current tobacco products in order to switch, but to still have nicotine and to still have the exposure, but reduce the risk of doing so. So the abuse potential needs to be carefully weighed for these types of products. Some of the challenges of nicotine pharmacokinetics and user behavior. The abuse potential is influenced by the rate and extent of nicotine exposure. So for example, inhalation leads to a very rapid delivery of nicotine into the lungs and brain, and that has a high abuse potential. When you have products that are delivering nicotine through other routes of administration like buccal, oral, or dermal, it's slower, it has lower exposure to your body, and therefore lower abuse potential. So a lot of the modifications can be made by simply changing the route of administration of a product. That's why you have things like nicotine gum that deliver the nicotine orally. PK, or pharmacokinetics, is influenced by user behavior and experience. So inexperienced users versus experienced users can have a very different amount of cigarettes that they inhale or products that they use, depending on their tolerability. And physical properties like particle sizes and other attributes to a particular product can influence how much exposure one gets when they're utilizing that product. So all of these have to be carefully considered when you're assessing and evaluating a given tobacco product. So when we're assessing abuse potential in different populations, we typically think about the types of individuals that we're bringing for these studies. And typically these are going to be experienced users. So by definition, these are people that smoke at least 10 cigarettes per day. They're going to be dependent and most likely population to switch to a new tobacco product. We have to think about, should discrimination testing be implemented, showing that they're enjoy the cigarettes and have a certain threshold of responding. The level of experience can influence the subjective rating. So a light user may not necessarily, may have a very different outlook and different experience to a product than somebody who has a very heavy habitual use of cigarettes. So being able to balance the population for appropriate responding and determining what the product is intended for and who to target in your clinical trial is very important. Usually these types of studies will have a positive control in which that is usually the subject's own brand of cigarettes that they smoke routinely. Non-users or former users are very important too, because the FDA and the regulatory agencies are concerned about people who have never used tobacco suddenly starting to use a new tobacco product, or people who have quit smoking tobacco reinstating their tobacco use on a product that they feel has less risk. So these populations also need to be considered in terms of how this new product may impact their behaviors in initiating or reinitiating tobacco use. For a general study design of abuse potential, and this is typically done in subjects that have tobacco experience, these are randomized crossover designs. Treatments include new tobacco products versus the usual tobacco product that's used by the subject. The study will include a screening period in which subjects are screened and selected for their suitability and eligibility for the study. And there's also a product acclimatization period. So if you're introducing a new electronic nicotine delivery device or a new device for tobacco, we want to ensure that the subjects are comfortable using it, know how to use it, and can get to a baseline of usability before we begin sessions, so that there's not a learning curve or an adjustment period and we control for that kind of behavior. So there are generally 2 sessions per treatment. You have a controlled session in which you administer a product. The smoking inhalation is defined and controlled. We'll give them a certain amount of time. We'll give them specific instructions on how to inhale, and it's conducted with a certain time frame. And oftentimes when you have smoking or vaping, you need to have controlled ventilated smoking chambers with an observation window. And these are critical in a study in order to be able to safely administer a vaped or smoked product and also protect staff from exposure. So we just talked about the controlled session. Then there is an ad lib session, which allows subjects to consume the product as much as they desire in a given time frame. And we monitor very carefully how much of the product that they're using. So this will be compared to a new product versus their typical standard product that they commonly use as a comparison. The study endpoints in these types of studies include various assessments that are subjective measures. So the subjects will be asked how much do they like the product, would they use the product again. It evaluates the different positive and negative effects of the product, and it may look for abstinence and withdrawal symptoms if they're not using the product. Other types of questionnaires can include the smoking urges, the modified cigarette evaluation questionnaire. We're also looking at various physiological components. We're looking at the pharmacokinetics and biomarkers, showing how much nicotine did they get exposed to. What are the effects on the physiology? Are there changes in the blood pressure or heart rate? And topography. Topography looks at the use of the product, the duration of the puff, the time between puffs, the frequency and volume of puffs. So how much of the product did they ingest in that sense? For smoking cessation studies, so there are tobacco products and nicotine-containing products that are intended to help people quit smoking. These require a longer study duration to assess behaviors. One needs to assess changes in behavior over time, showing that there is an actual reduction in the frequency of the amount of tobacco consumed, looking at the total days of abstinence, the reduction of tobacco usage, and looking at different physiological effects. So you want to look at reductions in the biomarkers, exposure and harm as a effect of reducing the amount of tobacco consumption. So these are studies that would require more in-depth analysis of the behavior of smoking cessation over time. There are also, in the guidances that I mentioned from the FDA, a lot of consideration about consumer perception. The FDA is very concerned about consumers misinterpreting the perceived labeling claims and having a false sense of safety or reduction of risk that may not necessarily be correct. So a lot of these types of studies will be centered around the product perception, bringing in market subjects to evaluate the marketing labels for these types of studies, for these types of cigarettes and products. It requires varying populations, ranging from non to heavy tobacco use, and those desiring to reduce, eliminate use, so that they can have a weigh in on what that package and what that label means to them. What do they interpret that as? You'll have varying demographics. You'll compare labels to marketed references, and there will be a variety of interview questionnaires to assess the comprehension of the label, what the subject perceives the risks of the labeling, and the likelihood to use that product based on the labeling. So in a recent FDA advisory meeting, and there have been several over the past few years, one product called very low nicotine, or VLN cigarettes, gave an application in just February of this year. In this meeting, the panelists agreed that the information about the product having low nicotine should not be withheld from the package or the labeling claims. There were, however, concerns raised for populations of people who never smoked and for adolescents, and the perceptions of safety that the product may have. It was recommended in this meeting that the information be added to the cigarettes to clarify that smokers should switch completely to the product in order to achieve the results similar to those obtained in the clinical literature. And the justification and the rationale behind that particular comment is that if the subjects continue to use higher risk tobacco products in conjunction with a safer or a product with reduced harm, they will not get that full benefit if they continue to use higher nicotine products or higher products with higher risk. So it needs to be made very clear that the data to support those claims are based on data where users were only exposed to that particular product. So that has to be, the conditions of which need to be clearly outlined in these labels. So now we'll switch a little to the biomarkers of exposure for tobacco products and talk about how we measure that in the clinical trial setting. So typically biomarkers are conducted for drugs. We look at the exposure and biological effects of drugs, but it is quite complex to evaluate these for tobacco products. The exposure, nicotine or nicotine equivalents. Nicotine has a very short half-life, so oftentimes you have to look for other metabolites to determine the extent of exposure. There are different chemicals in drugs, and there are different effects of exposure. Carbon monoxide, you have polycyclic aromatic hydrocarbons that are generated, aromatic amines. There are a variety of different chemicals that constitute exposure to tobacco products. Then you get into the biological effect. So the drugs, when you look at a standard drug product, it can have several biomarkers. It depends largely on the target of the drug. For tobacco products, you have a variety of different things that the products can exert biological effects on, including inflammation, cardiovascular effects, lipid profiles, lung function, DNA damage, genomic markers. There's quite a myriad of things to look at. For nicotine exposure, just to give you a frame of reference of how much nicotine is contained, an average tobacco rod contains between 10 and 14 milligrams of nicotine, and on average, about 1 to 1.5 of that's absorbed systemically during smoking. So it's a small fraction of what's actually contained. In most tobacco strains, nornicotine and N-ethtabine are the most abundant of the minor alkaloids, followed by anabasine. Nicotine and cotinine levels are influenced by many factors. So how much of it you get exposed to will depend a lot on the diet, meals, your age, sex, use of estrogen-containing hormone preparations for oral contraceptives, pregnancy, kidney disease, and other medications, and even smoking itself. The menthol in menthol cigarettes can impact the amount that you're exposed to. So a huge amount of factors and a lot of things that need to be controlled for when you're looking at it at these nicotine exposures in a clinical trial setting. When we develop biomarkers of exposure, we have to look at the analytical methods. Is the method specific, sensitive? Is it consistent? Can it be repeated reliably in a laboratory? And follow tobacco use patterns, you have to distinguish between tobacco users versus non-users. Does the assay distinguish between different types of tobacco products? Is there a dose-response relationship that corresponds with product use? So all of these things one has to consider when you're developing these biomarkers and validating them. It has to show that there's a difference between somebody who uses tobacco and somebody who doesn't. Otherwise, it may not be a sensitive and specific biomarker. In terms of measuring nicotine and some of the tobacco alkaloids, the nicotine has a very short half-life, means it gets metabolized very rapidly in the body. And so it has a very limited utility as a biomarker of tobacco exposure, particularly if you're looking at longer terms. So cotinine is a metabolite. It has a longer half-life with the bioanalytical assays. It comes in various matrices, so plasma, urine, saliva. It's used as a measure of daily cigarette consumption and secondhand smoke. It also shows exposure influenced by covariates affecting metabolism. The total nicotine equivalence is the sum of the nicotine, cotinine, the 3-hydroxycotinine, and their glucuronide conjugate. So you're looking at various different things to take in and sum up total nicotine equivalence. It accounts for differences in metabolism because people do have a lot of genetic variability with how they metabolize. And it has a stronger correlation with other biomarkers of exposures. The anabasine and antabanine, the monitoring abstinence or extent of active tobacco use during nicotine replacement therapy, these become very helpful for that purpose. So understanding what it is that you're trying to measure and the significance of it and what's the most appropriate is one of the critical tasks in identifying how you're going to assay for exposure in a clinical trial. Then there are other important markers. Carbon monoxide, for example, that's exhaled, the tobacco-specific nitrosamines. They have a longer half-life, so they can often be measured for longer periods of time, and things like polycyclic aromatic hydrocarbons that are exerted from tobacco. Also, not only do we look at biomarkers of exposure to tobacco and nicotine, we're also looking at biomarkers that may be predictors of disease risk. So, for example, so we're looking at biomarkers that can indicate potential harm. From some epidemiological evidence from the Shanghai cohort study, the urinary concentrations of the polyaromatic hydrocarbons, the total NNAL, and total cotinine are dose-dependently related to lung cancer risk. So they can be useful tools in determining how much of a risk there is. And for example, urinary concentrations of NN and the glucuronide are strongly associated with esophageal cancer. So again, there could be predictions of harm. If you look at some of the biomarkers of exposures in alternative tobacco products, smokeless tobacco, for example, there have been levels of higher carbon monoxide, PHS in smokeless tobacco users versus non-tobacco users, but these are lower than combustible tobacco users. In some ENDS products, forced switching from exclusive cigarette smoking to dual e-cigarettes reduce some of the biomarkers, as well as the THPs. When you looked at forced switching from combustible cigarettes, THS 2.2, reduced markers were also found in compared to the combustible cigarettes. So there have been studies to show that using these alternative tobacco products can reduce some of these biomarkers during exposure. When we look at the considerations and use and development of biomarkers, the Association with a smoking-related disease is important, what the risks are for cardiovascular, the different pulmonary disease, cancer. There's a clear relationship between the biomarker and early pathophysiology of disease. So the dose-response relationships correspond with smoking cessation or switching to a modified risk tobacco product. Pre-marketing studies must consider the time frame needed to see such a change. Oftentimes, the benefits and the risk of disease can take days, weeks, months before you can see noticeable improvements. And so therefore, clinical trials with such claims need to evaluate an appropriate time frame to substantially prove that there is a benefit and a reduced risk that is imposed by a product. There are also measurable and non-invasive collection methods that can be considered. Another quick comment, there was a recent FDA meeting on the use of an MRTPA. That meeting was held in January of 2018, and this was for an IQOS product that was shown to reduce the body's exposure to harmful or potentially harmful chemicals compared to combustibles. However, there was no support for the claim that the risk for tobacco-related diseases and switching completely presents less risk of harm than continuing to smoke cigarettes. So biomarkers of exposure appear adequate, but there may be more required for biomarkers related to reduced harm. Those are a little bit more harder to present because, again, you may need to have a longer study period to show that there are, in fact, reductions in harm. FDA also approved the first snus product in 2019. There were 8 products that were deemed then and given allowance to advertise with specific information about the lower risks of certain health effects using the products compared to smoking cigarettes. So this is now a more recent example where some risks were allowed for labeling claims, and using biomarkers, the Swedish Match US can market these products as with a claim that using general snus instead of cigarettes puts you at lower risk of mouth cancer, heart disease, lung cancer, stroke, emphysema, and chronic bronchitis. So this is now an example of how these biomarkers have evolved and how these studies have evolved to enable you to make claims provided that the data supports those claims. So in summary, the FDA guidelines continue to evolve following each product approval. Assessment of these new tobacco products requires clinical evaluation, including abuse potential assessment. It depends on what it is that you're trying to claim and what your product has been designed to do. And so that has to be critically evaluated and strategically developed in a way where you're conducting trials that are relevant and that are going to have meaningful endpoints to support justifiable labeling claims. Biomarkers can provide critical data to support claims of reduced harm. Robust studies are needed. You're going to need to have strong data that shows those claims of reduced harm or reduced risk. Addressing the consumer perceptions and risks in various population subtypes is critical, and that's a common theme across all of the guidances in the FDA. So I'd like to thank you for your attention today, and I'm happy to answer any questions. Thank you.